Zoology, and since we had discussed the basic parts of your cell, their organelles, the processes that happens inside your cell and outside, we call it the intracellular and extracellular activities, and the different types of tissues that constitutes different types of organs, and all of the organ systems of your human body or the animal body. So now we will be going in depth into the cell division and inheritance. So humans have known for centuries the traits that are inherited from their offspring to their parents. So we have manipulated trial and errors for breeding to achieve our desired characteristics for the animals, because it was being experienced through time that animals, plants have been crossbred with each other, so that the desired results that we're getting is attained through crossbreeding these individuals. But the question is, how did it happen? Okay, so what are the molecules that are responsible for doing this? What are the materials that are controlling the inheritance? And what are the positive and negative impacts of human interacting with animal reproduction? So we'll talk about this one in the future slides. So reproduction is essential for life. Each organism is existing solely because of the ancestors succeeded in producing progeny, or what we call as the offsprings, okay, that could develop, survive, and reach reproductive age. So life has to continue because of reproduction. So for organisms that are composed of only one cell, we call that unicellular organism. Cell reproduction is also reproducing the entire organism. For multicellular organism, the cellular organisms are involved in reproduction, such as the repair, the growth and production of sperm and egg cells, so that the animal are able to reproduce. So genetics is an emerging field that was a study of how the biological information is being transmitted from one form or one generation to the next. Okay, so this is all about the mighty DNA. Okay, the recently discussed DNA, that it has the ability to replicate, and it is able to carry down the information from the offspring, okay, and their parents, and vice versa. Okay, so that's the transfer of traits that are important in reproduction. So what about eukaryotic chromosomes? So the focus of zoology is to study the eukaryotes, okay, but the main focus does not rely on all the eukaryotes, since we are dealing with the kingdom Animalia, and eukaryotes are special for their membrane-bound organelles and the nucleus that has surrounded by a membrane. Okay, so we will talk about the chromosomes inside your nucleus, where it stores the genes. Okay, we call this one as the units of inheritance. They are actively participating in the formation of your proteins. So think about it, like DNA is what instructs, okay, the cell to release, okay, proteins. The DNA is like the blueprint of life. It varies from animals, plants, and all the living organisms on Earth. So the cell's chromosomes are highly dispersed. Okay, so there are some highly dispersed state, we call them chromatin because they are colored bodies, as it was discussed in the cell tissues organ systems presentation. So the chromatin is consisting of DNA and histone proteins, and the association of DNA and protein with the complex jobs in condensation, okay, and regulating DNA activity. So the chromatin is DNA plus histone proteins. The DNA is surrounding your histone proteins. And we have 5 different histone proteins. The amino acid composition of these proteins creates a positive charges that attract the negative charge of the DNA's phosphate groups. Some of these proteins have a core particle, and DNA wraps coil around the proteins, a combination what we call as the nucleosomes. Okay, so for us to understand the the organization of your chromatin and the DNA and the chromosomes, we will be viewing a figure later on. So not all chromatin are active. For human genes, for instance, they are active only after adolescence. Okay. In other cases, entire chromosomes may not function in particular cells. Inactive portions of your chromosomes produce dark banding patterns with certain staining procedures. We call that heterochromatic regions because they are the stained or dark banding regions, whereas active portions of chromosomes are called the euchromatic regions. So this is your organization of your eukaryotic chromosome. We'll start with the basic. Okay, below we have the DNA, duplex matter, and then it coils around the nucleosome. Okay, so we call that the histone core in the middle, the proteins, and then it will become a solenoid. The solenoid will create a chromatin loop with scaffold proteins, which is the scaffold proteins are maintaining the shape of the chromatin loops or the rosettes of chromatin loops, which are found in your chromosome. So this is your chromosome consisting of a long DNA molecules that wrap histone proteins. Okay. So this is what we call as the arms. Okay, the P arm and the Q arm, and all of the information regarding the chromosome are being discussed in your genetics. So what are sex chromosomes and autosomes? When we say sex chromosomes, these are chromosomes, okay, that depicts the sex, okay, of an organism. So in order for us to discover this one in the early 1900s, attention turned into the cell to find a chromosomal explanation for the determination of the maleness or the femaleness of an organism. So some of the evidence on a chromosomal basis for sex determination come from the insect proteinor. Okay, this is the insect proteinor. Only darkly staining chromosome of proteinor called the X chromosome is represented differently in males and females. So all of the body cells, or say somatic cells, which are the kind of cells that are responsible for growth and repair, okay, are noted by 1X for the male chromosome. Okay, look at this one. All somatic body cells of males have 1X chromosome, and all the somatic males of females have the 2X chromosome. So we call it XX. Same thing with humans. We have 2X chromosomes for the females and 1X for the male and 1Y for the male. Similarly, half of all the sperm contain X and half contain no X. So when the proteinor's chromosome is read as XO, it is in the male, but for female it is double X or the female proteinor. So this pattern suggests that fertilization involving the X-bearing sperm will result in female offspring when they are fertilized, and that fertilization involving a sperm with no X chromosome will result in a male offspring. So this is the figure 3.2, which illustrates the sex determination system in a 50-50 ratio for males in these insect species. Okay. Chromosomes are also represented differently in females than in males, and the function of the sex determination are sex chromosomes. Chromosomes that are alike and not involved in determining sex are called the autosomes, okay, or the somatic cells. This is your XO system of sex determination for the insect protodor. So this is just a an illustration for the males and the females, and then they come to fertilize. When the X has not received another X chromosome, it is considered male, and the other one is a female when there's two X chromosomes received by the zygote. Okay. So for other animals, okay, like humans and fruit flies, we have the XY system of the sex determination. In the XY system, males and females have an equal number of chromosomes, but males are usually XY, and the female is usually XX. In birds, the sex chromosomes are designated for ZW. The female is the ZW. It's the opposite, okay? Even though the X and Y chromosomes are called sex chromosomes, they also help determine non-sex related characteristics. Okay, it is true for the X chromosome of most animals. Okay, so the common X chromosome for the most animals is very large and has genes that code for many traits. Since autosomal chromosomes frequently carry genes that influence sexual characteristics, this mode of sexual determination also results in approximately equal numbers of male and female offspring. So we have number of chromosomes. Okay, so the number of chromosome varies with within the different species. Okay, so chromosome number vary greatly among these individuals. So usually the chromosome of your animals will range in the 10 to 50. So they are present also in sets. The number of in a set being a characteristic of each kind of animal is expressed as n. Okay, so when you see n, it identifies the different kinds of chromosomes. But for most animals, we have two sets. Okay, so the set is n, so we call it the 2n chromosomes. This is the diploid. Okay, when you say double condition, some animals have only one set, so we call them n chromosomes, and they are what we call as the single or haploid. Examples are honeybees or male honeybees and some rotifers. But some of the animals, okay, have more than diploid numbers, so they have poly, okay, or more chromosomes or more sets of chromosomes. We call that polyploids. So the process is polyploidy, or a condition. The upset of numbers in sex chromosomes apparently interferes with reproductive success. So asexual reproduction often accompanies polyploidy because there's a lot of zygotes that are there's a lot of sperm and egg has been fertilized, and when you say the brine shrimp, let's say for instance, released, okay, millions and thousands of fertilized zygote in the water, not all of them will survive. Okay, so it also interferes with reproductive success. So we'll talk about the cell cycle and your mitotic cell division. So what are these? Your cell has to commit its existence. Okay, also there's a life cycle of a cell. Same with all the living entities. Cell must begin with a parent cell, and then to produce the new cell, then it goes a maintenance and growth process until it matures, and ultimately it produces another generation of two cells, etc. So the cell cycle is a complex process wherein all the eukaryotic cells undergo this stage. Okay, so this is the stages of your cell cycle: the G1, okay, the G2, and the S and the M phase. So what are these phases? The G1, S, and G2 is the longest process of your cell cycle, so we call that one as the interphase. Okay, 70% of 80% of the entire cell cycle will be allotted for the interphase. Okay, so what happens during the S phase, G1 and G2 phase, is that the cells are they are fully examined. They are examined if they are capable of multiplying or dividing in the M phase. So the G1 phase will check all the necessary processes like their metabolism, the growth, until a certain such a point where they can pass through all these stages. When they aren't able to pass the stages, they undergo in the G0 phase. Okay, when they are completely out of the cycle, they are being removed from the cycle. Okay, so the length of the different stages varies greatly from one cell to the next. So once they undergo the three sub stages of your interface, they will successfully undergo the M phase, which is the mitosis. Okay, so what happens, sir, if the process will not, the examination will not prosper, and there are faults that are being passed, okay, within the three sub stages, and then it underwent the M phase. The science of cancer is based on this cycle. So there are some abnormalities within the cell that they are reproducing the abnormal cells. So cancer might occur. So that's mutation technically. So we have the G1 or the first growth, second growth, and the M phase. So after the M phase, we call the process a cytokinesis, wherein the cytoplasm is being divided into two different portions. So we have the chromosome replication in homologous chromosomes. In this example, we have the centromeres in the middle. This is where the kinetochores are attached, okay, during the pulling over of your sister chromatids away from each other during the Anaphase stage. So we will talk about that one later on. So we call them homologous chromosomes because they are all at the same length and it goes for the same gene. Okay, we say homologous. So we call this replication process is the blue one comes from the father and the red one is from the mother. The paternal and the maternal chromosomes are duplicating, okay, in your G1 phase. So before replication, chromosomes consist of a single chromatid. So they will call the one chromatid, and when they have already replicated, we call them the sister chromatids. So we'll talk about interface. Each chromatid attaches to the other copy or sister at the point of constriction. We call that centromere, and the centromere is a specific DNA sequence of about 220 nucleotides and has a specific location in any given chromosome. So bound to each centromere is a disc protein we call as the kinetochore, which will be the attachment sites of your mitotic spindle. So in the M phase or mitosis, we have 5 different phases. Okay, the prophase, prometaphase, metaphase, anaphase, and telophase. So in a dividing cell, however, the process is actually continuous, with each phase smoothly flowing to the next. So the first phase of your mitosis is called the prophase. So that's before phase. So it your chromosomes there will begin to be visible, okay, when you subject it to the light microscope. They are like thread-like structures, okay. The nucleoli and the nuclear envelope begin to break up, and two centriole pairs move apart. So the centrioles are just following on the opposite poles of your cell. So by the end of the prophase, the centriole pairs at the opposite pole of the cell, and then they will radiate this array of microtubules. They will spread it out, and then we call that asters or little stars, which attach to each centriole against the plasma membrane. So between the centrioles, the microtubules form as spindle fibers, which extends from pole to pole, and these asters, spindle centrioles, and microtubules are collectively called your mitotic spindle or your mitotic apparatus. So this is the continuum of your mitosis in cytokinesis in a figure. So these are your microtubule organizing centers. They are pushing away each other, going to the opposite side of the cell. They release asters. They will attach the spindle fibers to the centromere of your sister chromatid. They will pull them apart away from each other in the anaphase. Then they will be attaching each other from the opposite sides of your opposite hemispheres, okay, of your cell. Then they will form a cleavage furrow. Once the cleavage furrow is formed, it undergo a telophase, and then we will be performing the cytokinesis, which the division of your cytoplasm. So we have two daughter cells now. So again, this is just a review of the phases of your mitosis. So during the metaphase, once again, they will be aligning in the middle part of your cell. In your anaphase, they will ana, which means back again phase. So they will be shortening the microtubules and pulling away from each other. In the telophase, mitosis is over, but cell division is not. Okay. So the telophase is the last phase of your mitosis, where the mitotic spindle disassembles. So that is a preparation with another type of cell division, another round of cell division. And lastly, the cytokinesis. The furrow will deepen. It will be creating a cleavage, and then until it loses the two daughter cells. So what about meiosis? For mitosis generally releases somatic cells or identical cells. That's when you have bruises or lacerations, you have wound, or your internal organs are wounded. The body will repair itself or will heal itself through the process of cell division, releasing somatic cells. So they should be identical in nature. But for meiosis, these are the bases, or these are the sex cells that are being reproduced by your body. And it requires the genetic contribution of two different sex cells. And these two different sex cells are the egg and the sperm. We call them gametes. Okay, gametes because it's like wife and the husband. Okay, so a male gamete is the sperm and the egg is the female gamete. So they will fertilize and form a zygote. Okay, zygote, which means yolk together. So the zygote is the first cell of your new animal, and the fusion of the nuclei within the zygote brings together genetic information from the two parents. So the parents are contributing to the entirety of the zygote. So from the mother side, 50-50, okay, of their genetic information has been released. So to maintain a constant number of your chromosomes in the next generation, they have to reproduce sexually, okay, and producing the gametes with half the chromosome number of their ordinary body cells. So the ordinary body cells, as I say, are called the somatic cells. So all of the cells in your bodies, except for the sex cells, are called somatic cells. Okay. So all of the cells, the human body, or the most animals, except for the egg and the sperm, have a diploid number of chromosomes, or the 2n. And gametes are produced by cells set aside for that purpose early in development. So these cells are called the germline cells, and they undergo a cell division. We call that meiosis, or the diminution. So the Greek word for meiosis means diminution. Okay, diminution. So what is we have the meiosis, and then it's divided into two different phases. The first meiotic division and the second meiotic division. So we'll talk about the first meiotic division. Okay, you familiar with the chromatin? Okay, it folds and chromosomes become more visible. So just like the mitosis, because a cell has a copy of each type of the chromosome from its original parent cell, it contains a diploid number. So we can see the homologous chromosomes there. Okay, they carry genes for same traits, and they have the same length and similar staining pattern. Okay, we can see that one in your when we try to stain the chromosomes, and then they are aligned. Okay, and after the staining pattern, we could identify that they are similar. Okay, they have the same length. They are identified as matching pairs. So during the prophase, one homologous chromosomes line up side by side in a process called synapsis. So the synapsis will let the meiotic products as different from their parents. Okay, so they will form a tetrad of chromatids, also called as the bivalent. So this is what your meiosis one looks like. So this is just like the same of mitosis, but homologous pairing is of your chromosomes is existing in this part. Okay, so the red one is for your mother, the blue one is from your father. So they exchange the materials, okay, of your chromosomes, producing the nuclear envelope during the telophase one, and then we have the daughter cells. So almost the same with your mitosis. Okay. So the tetrad contains two homologous chromosomes. One is maternal in origin, and one is paternal in origin. So synapsis also initiates the crossing over, okay, whereby the ancestor chromatids of two homologous chromosomes in a tetrad, they exchange the DNA segments. Okay. So this is facilitating crossing over, which is a form of genetic recombination, which makes all the animals in varied forms. Okay, varied. They have varied genes. They have varied morphology. That's why the diversity of life is very unique. It's because of the sexual reproduction. Okay. So this is your synapsis and the crossing over. It's a very tight gene-to-gene pairing of the homologous chromosomes during the prophase one of your meiosis. So what about the second meiotic division? So it's resembling like an ordinary mitotic division, except that the number of chromosomes has been reduced. Okay, if your mitosis will produce two daughter cells, here it is being reduced by half. The phase of the second meiotic division are the prophase 2, met 2, anaphase 2, and telophase 2. At the end of your telophase 2, in cytokinesis, the final products of these divisions are 4 new division products. So most of your animals, each of these division products is haploid. We call that a haploid because they consist of one set of chromosome and may function directly as a gamete, okay, or a sex cell. So this is your second division of your meiosis. Try to look at the same process. The centrioles will release the asters to get around with the opposite poles of your cell. Centromeres are located in the middle where the microtubules are attaching. They separate during the anaphase 2, and then they will be enclosed by a nuclear envelope, okay, and the sex cells are being released. So try to see what happens during this time. They have during the prophase 2, this one pair of chromatid, or the sister chromatid, has already a part or a little part of the other chromosome. So when they pull apart with each other, they have unique sets of traits or genes coming from the parents. Okay, so like these are the sex cells or the gametes. So these are the sibling cells. Okay. So what about releasing the sex cells? Okay, for the males we have spermatogenesis, and for the females are oogenesis. So for most animals, okay, the results of meiosis will form the sperm and the egg cells. So the spermatogenesis produces mature sperm cells and follows the sequence previously described. And all products of meiosis require flagellum for locomotion. So the flagellum, if you can still remember, it's used for locomotion for the sperm cell. It's quite long structure, okay, for movement, and a cap-like structure on the head of your sperm to penetrate the egg. In oogenesis, it produces a mature ovum or egg. So it differs from spermatogenesis in that only 4 of meiotic products develops into a functional gamete. Okay, so the products of meiosis are called polar bodies and eventually disintegrate. So for oogenesis, out of 4, only 1 will be functional, and the other ones will the 3 other more will disintegrate. So in some animals, the mature egg is the product of first meiotic division and only completes meiosis if it is fertilized by your sperm cell. So what about DNA? Okay, we'll talk about this in the first few slides. The DNA is your genetic material, or the deoxyribonucleic acid, which is discovered by Watson and Crick. So we have 3 characteristics on how we explain life. Okay, so the 21st century biologists have studied this molecule, we call that DNA, and it must have certain characteristics to explain how life emerged, or how an organism is being called a living organism. First is that genetic material must be able to code for sequence of amino acids. So that's the blueprint of life. So if your DNA has been creating glycine, okay, it's creating different sets of amino acids, then it must be a living molecule. Okay, it will be coding for life. Second, it must be able to replicate itself. So during the S phase of your cell cycle, DNA is replicating itself. So from parent strand, they will produce the daughter strands. And third, it must be in the nuclei of your eukaryotic cells. Yes, of course, because DNA, okay, you can remember the eukaryotic chromosome, the DNA is surrounding your histone proteins. So in order for them to create a solenoid, rosette of chromatid loops and a chromosome, they must be surrounded, okay, with the histone is surrounding, that the DNA will surround your histones. So the double helix model, okay, is the structure of your DNA. Okay, so we have 2 kinds of molecules which is participating in protein synthesis. Okay, the 2 kinds of molecules is the DNA. The first is the DNA, the genetic material, and the other is the ribonucleic acids, or the RNA. It's produced in the nucleus and moved into the cytoplasm, where it participates in your protein synthesis process. So this area of our study is what we call as the molecular genetics. We will talk about the molecules that are participating in reproduction. So both the DNA and the RNA are large molecules. We call them nucleotides. Okay, so a nucleotide is a nitrogen-containing organic base in the form of either a double ring or a single ring. So for a double ring, we call that purines, and a single ring is a pyrimidine. Okay, so nucleotides are also containing a pentose, or a 5-carbon sugar, and a phosphate, or 2 phosphate groups. The DNA and the RNA molecules, however, differs in several ways. The DNA and RNA contain the purine bases adenine and guanine, and the pyrimidine base cytosine. Okay, the second pyrimidine in DNA, however, is thymine, whereas in your RNA it's different. It's a uracil. Okay, so we'll talk about the base pairs. Okay, so this is the purines. Okay, adenine, guanine, the pyrimidine, cytosine, uracil, thymine. So the base pairs are adenine and thymine for the DNA, okay, and guanine and cytosine, all. And for the RNA, okay, it's guanine and cytosine, but instead of adenine and thymine, for RNA it's adenine and uracil. Okay, so these nitrogenous base form, okay, a tight bond with each other. Okay, these bonds will help the DNA for its double helix shape. Okay, so remember the sugars or the carbohydrate groups in your DNA is a deoxyribose, whereas in the RNA is a ribose. So a ribose, a hydroxyl group of OH would replace the hydrogen shaded purple. This one. So the double helix model is not a twisted ladder. So maybe if you have underwent a model activity in your high school, in your grade school, you will be asked to create a model of your DNA. It's like a straw model, like you pair the ATCG, then you twist the ladder. No, it's not really the the model of your DNA. If you have seen or have passed through a helical staircase, okay, a spiral staircase, that's the model of your DNA. Okay, it's ladder like, but it's a twisted a spiral staircase. Okay, so the nucleotides, as I said earlier, that attached by a strong bond, we call that covalent bond. Okay, between the phosphate of one nucleotide and the third carbon of your deoxyribose. So this one are the base pairs, ATCG, for your DNA. So for double helix model also, they have opposite in polarity, and the DNA molecule is said to be antiparallel. So they are twisted to a right-handed helix with one complete spiral every 10 base pairs. So this is your structure of your DNA. So nucleotides of one strand of nucleic acid joined by linking a phosphate of one nucleotide to the 3' carbon of an adjacent molecule. Okay, so it works on 5' to 3' and then 3' to 5', so that they will be creating this 2 nanometer distance, okay, between the two strands, and 3.4 nanometer for this this base pair until here. Okay, so it forms this double helix structure. The curved arrows here is the three-dimensional representation of your DNA. So the curved arrows will represent the spiral antiparallel nature. So what about DNA replication? In eukaryotes, okay, the DNA strand is a template for a new strand. We call it parental strand, okay, and the base pairs are required, or the base pairs will dictate the positioning of a nucleotide of a new strand. Thus, each new DNA molecule contains half of the old molecules, which is conserved with the new molecule. So DNA replication is said to be semi-conservative. So this is the example. So for the original double helix, so this one is the daughter helices. Okay, this is your parental strand. So of course they will be passing through. The one is leading and the one is lagging. Okay, we say leading. This one. Okay, the strands will separate and complementary base align opposite templates. Okay, and then enzymes link sugar phosphate elements in aligned nucleotides into continuous strands. So we have daughter helices and creating the new strands of your DNA. Okay, so what happens during this time? So only one on this example, there's only one direction shown here for simplicity, and then the double helix will unwind. Okay, so the double helix will pass through here to unwind the DNA structure, and then the original strand will serve as a template for a new strand, and other enzymes such as the DNA polymerase will help, okay, the nucleotides with exposed and paired base on the wounded portions of your original DNA, and link number 4 is the nucleotides into continuous strands like the ligase. So we have genes in action. The other processes of your protein synthesis is undergoing the same process. We call this transcription and translation. Okay, so genes can be divided in a sequence of bases in the DNA that goes for synthesis of one polypeptide, and genes must somehow transmit their information going to cytoplasm. So the synthesis of RNA molecule from a DNA strand is what we call as the transcription, or the scribe, okay, to write, okay, the DNA info going to the RNA info. And the formation of protein from RNA is what we call the translate. Okay, translate the mRNA codes or the codons going to create an amino acid. And amino acids are a series of polypeptide that will create your proteins.